To start a muscle contraction, first the action potential must travel down the motor neuron. Action potentials are impulses that travel from our brains or our spinal cord to trigger the contraction of a skeletal muscle. When the axon of the nerve meets the muscular cell, this is called a neuromuscular junction. This is the point that the axon terminals of the motor neuron touch the motor end plate of the muscle fiber. Because the action potential is electrically charged, it causes voltage-gated calcium channels to open, and the calcium actually goes into the axon terminal. In this terminal, there is sympathetic vesicles, and inside these contain acetylcholine, which is abbreviated as ACH. Those are right here. Right here, these blue things are the ACH receptors that when the calcium channels open up, they cause the sympathetic vesicles to release the acetylcholine that binds with the receptors. The acetylcholine diffuses into a sympathetic cleft, which is the space between the axon terminal and the muscle fiber. When the AC binds with the receptor, it opens these ligand gates and ligand channels and allows sodium into the cell. A action potential passes through the sarcolemma, which is this right here, and causes the voltage-gated sodium channels to open up, and the sodium ions enter the muscle fiber. So they enter the muscle fiber, and potassium exits the muscle fiber. The more positively charged in the muscle fiber, the more gates will open up. When the sodium rushes into the sarcolemma, it depolarizes it. The sarcolemma is the plasma membrane that goes around the muscle fiber. In this step, is when excitation contraction coupling starts. The action potential travels along the sarcolemma until it reaches the T tubule, which is a hole that goes down into the muscle fiber. The action potential uses T tubules because the signal needs to get deep enough into the muscle fiber. These gates are voltage gated because a charge makes the channels open. The calcium released from the sacroplasmic reticulum reaches the myofibril, which contains myofilaments. So the myofilaments are proteins called actin and myosin. The sarcomere contains the thin actin filament and the thick myosin filament. When calcium levels rise, the calcium ions start to bind with the troponin. Troponin is attached to the actin filament, and when it binds with calcium, the shape actually changes, which removes the blocking action of the tropomyosin from the actin binding sites. So this allows the myosin head to bind with the actin and form a cross bridge. Before this cross bridge can occur, though, the myosin head has to be activated. When the ATP binds to the myosin head, it must hydrolyze into ADP and an inorganic phosphate. So the energy from the ATP activates the myosin head to go back to its original position. So when the myosin attaches to actin, the phosphate is released, and the bond between the actin and the myosin becomes stronger. So When the ATP is released, it activates the myosin head to pivot, which causes the actin filament to slide forward and towards the center of the sarcomere. The cross bridge detaches when the ATP binds to the myosin head. The bond between the actin and myosin then weakens, and the myosin head then it detaches. As long as the actin binding sites are exposed, the cross bridge cycle will repeat and muscle contractions will continue. So during the cross bridge cycle, the actin filaments get pulled towards each other and the sarcomere shortens. So this is relaxed and this is contracted. The cross bridge cycle ends when the calcium ions are transported back into the sarcoplasmic reticulum. The troponin, this returns back to its original shape and allows the tropomyosin to cover the myosin binding sites on the actin filament. This allows the muscle to stop contracting. When you have a disease like myasthenia gravis, your immune system mistakenly attacks your muscles. So it's actually interrupted at the neuromuscular junction. Their body destroys or blocks the receptors for the ACH, these receptors right here, which prevents the muscle from contracting.